Welcome, Luca. Thank you so much. So Luca Casula is from the University of Cagliari and is part of the work package on prodrugs and formulation. So he will speak now about the formulation approach. Thank you.
Thank you for the kind introduction. So I am Luca Casula, and I'm a postdoc fellow at the University of Cagliari in the group of nanotechnology for drug delivery. What we do in our group, we try to develop proper formulations to improve the delivery of drugs that have solubility problems or bioavailability problems. So some of the techniques that we use are nanosuspensions or liposomes, which have become famous during the COVID pandemic because some of the vaccines were prepared using liposomes, but also nanofibers or microneedles. We focus on the oral delivery, but also skin delivery, pulmonary delivery for some diseases such as asthma or BPCO. And we also have some active project for brain delivery on Parkinson's disease. So the aim of our group in this project was to develop proper formulations for a more effective absorption of one of the prodrugs that was synthesized by the University of Cardiff. And the first choice in our strategy was the development of a stable nanosuspension. Why a nanosuspension and what is a nanosuspension? Nanosuspension can be defined as a carrier-free nanoparticle of pure drug. What does that mean? It means that there is just a drug with a stabilizer. There are no other excipients. So we are avoiding any adverse reaction. And why nanosuspension? Think about what happens when you put sugar in water. Sugar is going to dissolve, yes, but it takes some time. You need to stir it or to eat the water. Why? Because the crystal of sugar can be the bigger cube here. It is surrounded by water only in some surfaces. If you cut the crystal into smaller crystals, then the water surrounding the surface will be more. That's what we are doing with drugs. We're increasing the particle surface area to increase also dissolution velocity and saturation solubility. Here, just trying to be very simple, how we produce our nanosuspension through a milling technique. So we have milling beads that are going to disrupt the crystals of drug to obtain smaller crystals and a stable nanosuspension. This is the technique that we usually imply in our labs. However, the prodrugs that we used, synthesized by the unit of Cardiff, has a very high hydrophobic character. So what happened here is that what you can see here on the back, you can see the milling bits, and I hope you can see it, on the top there are drug aggregates. So this is because of the high hydrophobic character. So the bits were not able to disrupt the particles in order to obtain a stable nanosuspension. So the wet ball media milling was not a proper technique for this study. So we had to think about new strategies. So we try to optimize a different technique, such as a bottom-up technique to obtain nanosuspension, but also we wanted to try new techniques to obtain a different formulation, such as a polymeric heavy ball film. So very briefly, this was the technique that we used to produce the nanosuspension through a solvent-anti-solvent precipitation. And what we obtained was a homogeneous nanosuspension. As you can see here, the picture on the left, it has a very different aspect than the picture that I showed you before. So we can see here that it's completely homogeneous. And from the analysis we obtained, we saw that the drug crystals had a size diameter in the nanoscale and were also homogeneous. But the most important information here is that we carried out solubility experiments, and as you can see, the solubility of the drug when formulated as a nanosuspension was almost twofold higher than the raw drug. So this could be very important in developing the formulation and improving the absorption of the drug. So what are the possible application of the nanosuspension formulated? It could be administered as orally, has a dry powder, so through a lyophilization process, and of course some redispersibility studies, being encapsulated in gastro-resistant capsules in order to avoid any possible degradation in the stomach by the acidic environment and be absorbed in the intestine. Or another option could be the local administration using needle-free jet injector. Because as my colleagues told before, everything that is administered orally goes through the first pass metabolism. That is, might reduce the amount of drug that is going to the active site. So if we want to avoid that, we could use such a device. What is a needle-free jet injector? It's a medical device that is going to create a micro hole on your skin through a high-speed stream of fluid of the formulation caused by the action of the spring, and it is needle-free, so mostly painless, and the drug, the solution or the formulation can be delivered at different levels, so intradermally, subcutaneously, or intramuscularly. And these kind of devices have also been used during the COVID pandemic here in Italy. They were approved for vaccines. So the main advantage, of course, is the user-friendly injection, because you do not need a nurse or a doctor to give you injections. Elimination of the risk of needle stick injuries, and of course, increase of compliance in needle-phobic patients. The question now is, are these kind of devices compatible with the formulation we produce with nanosuspension? The answer is yes, because we carried out already a study about that. And what you can see here is a microscopic image of the skin. On the top, you can see the hole that is created by a classic needle. On the bottom, the one created by the needle-free jet injector. And what is also important is that I told you before that what is important in nanosuspension is the diameter of the crystals. Are we affecting the diameter of the nanocrystal? The answer is no, because as you can see here from this graph, the size of the crystals was not affected. And also the release of the drug, here we used a subcutaneous model, the release of the drug when injected using this kind of devices or a classic syringe is the same, is comparable. So the two strategies, the first one was to produce nanosuspension with a different technique, and I showed you the data. The second one was to optimize a new technique to produce a polymeric edible film. What we thought about, we are kind of specialists in nanofibers, which are polymeric solid fibers with nanoscale diameter. Here you can see the microscopic aspect of this kind of formulation. But as I was telling you before, for the crystals, the smaller, the more water can go through them. It's the same for the nanofibers. More water or more biological fluids can go through the fibers and enhance the release of the drugs. Very simply, nanofibers are produced through polymeric solution and application of an electric field. But what is important here is that we were able to obtain nanofibers incorporating the drug. This is what we have at the first steps of the production of the drug. It's like a spider web with a fibrous structure. But the more you produce the drug, the more thicker it becomes. And it is really like a polymeric strip, edible polymeric strip that you can administer orally. We managed to use a gastro-resistant polymer. So it means, again, that we are protecting the drug from the acidic environment of the stomach, and the drug will be released only in the intestine. It is maintaining the fibrous structure that is helping the release of the drug. And also it has an adjustable dose. It means that the bigger or the smaller you cut the polymeric film, the less or the more drug you will have. And also you can also adjust the polymeric film, and if the patient doesn't want to have this administered, you can put it into capsules and just administer as a normal capsule. So I would like to thank all the colleagues that took part in this project, the University of Cardiff for sending the Prodrugs, and all the organizing committee for this amazing event. Thank you.